Hey everyone, Dr. D here, and in this video we are going to be doing an Exam 3 review for Biology 2420 at TCC. This exam covers chapters 12, 13, 15, and 16. So let's go ahead and get started.
Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Dr. D, Dr. D, Dr. D. Explain stuff.
Now, as always, please study all your handouts to be really properly prepared for the exam. This review video is going to highlight some of the most important points that you need to know to do well. So let's go ahead and get started with Chapter 12, covering antimicrobial treatment. Please be aware of the characteristics of an ideal antimicrobial drug. Here they are listed, and here are some more. Obviously, please know the terms in bold. I'm not going to spell out all of them, but if there's a term in bold, it most likely is an important term to know. All right, please know the difference between semi-synthetic drugs and synthetic drugs. Remember the Kirby-Bauer test, the disc diffusion test, and how you measure the zone of inhibition in millimeters? We did that in the lab as well. And remember, based on the size of the zone of inhibition, and these charts, you're able to determine whether a bacterium is resistant, intermediate, or sensitive to a drug. Remember what the minimum inhibitory concentration, or MIC, is. You should remember that. It's the smallest concentration or dilution of drug that visibly inhibits growth. This is an important concept. Remember the therapeutic index, or TI? It's the ratio of the dose of the drug that is toxic to humans to its effective dose. Remember, the larger the TI, the better the drug. Remember, the lower the TI, the riskier the choice. So remember that a larger therapeutic index is best. Obviously, know the difference between broad spectrum and narrow spectrum drugs. Remember that penicillin and the other penicillins, like these listed here, remember that they have a beta-lactam ring. That's this ring right here, this ring shape right here. Remember that that ring is important because it's the target of penicillinase. The bacteria that are resistant to penicillin, they form a enzyme called penicillinase that destroys this beta-lactam ring. Isn't that interesting? Another thing you should remember is that some antibiotics target the cell wall, and you should know that penicillins target the cell wall. It's very important to remember. And then please remember that some antibiotics target protein synthesis. Remember, they prevent the bacteria from making proteins, usually by interacting with and blocking the ribosomes. Remember, the ribosomes are the little organelles that synthesize proteins. Okay, and the class that I want you to remember that targets protein synthesis are called the tetracyclines. So you see, the drugs that target protein synthesis, tetracyclines. So let's remember that tetracycline is an example of a drug or antibiotic that stops protein synthesis. And then there are also antibiotics that inhibit certain metabolisms, such as folic acid synthesis. And I want you to know that it's these sulfa drugs or sulfonamides. These are the ones that target folic acid synthesis. So we should know that. All right, there are also antibiotics that target DNA or RNA replication. And what you should know is that quinolones are an example of that. This is really important to understand. Remember I told you about fungal infections? I told you that because fungi are eukaryotes like you and me, that means that this presents problems for treatment because of that similarity, that evolutionary similarity. A lot of drug targets against fungi also affect you and humans, right? So this means that they tend to have a low TI or therapeutic index. See, similarities between fungal and human cells mean that drugs toxic to fungi are also capable of harming human tissues. This is why it's so hard to treat fungal infections. So we need to know that. Also, we need to know about antiviral agents or antiviral drugs. Do you remember that most of these antivirals, they don't actually cure the infection. They simply slow down the infection, slow down the viral replication, and it's really usually up to your own body to cure or pass the virus or not. Next, how do these microbes become resistant to drugs? So these are known as mechanisms of drug resistance. So for example, do you remember penicillinase? Penicillin-resistant microbes can form this penicillinase enzyme that cleaves the beta-lactam ring of penicillin, inactivating the drug. Okay, so that's an example of, a enzyme that is synthesized that inactivates the drug. This requires that the organism have the gene to produce that enzyme. Another mechanism has to do with permeability or uptake of the drug. So if you can prevent the drug from being taken up by the microorganism, that's another way to limit the potency of a drug. The drug is immediately eliminated. For example, the microbe could form a pump and pump the drug right back out if it enters the cell. Mutation could occur. Mutations can occur that affect the binding sites of a drug to the enzyme or the protein. And for metabolic pathway inhibitors such as sulfonamides, for example, the effective pathway is shut down, but an alternative pathway could be used to do the same metabolic pathway or the same metabolic result. Do you remember how microbes can become resistant with time? This, remember, happens when lots of bacteria are present, but only a few are drug resistant due to random mutation. The antibiotics kill most of the bacteria causing the illness, but leave the most resistant bacteria behind, alive. Those resistant microbes grow and proliferate, and they become the majority of the population. And then later on, through horizontal gene transfer, those bacterium can give that information, that drug resistant, to other species of bacterium as well. Please know the difference between probiotics, which are preparations of live microorganisms fed to animals and humans to improve intestinal biota, and be able to compare that to prebiotics, which are not organisms. These are nutrients that encourage the growth of beneficial microbes in the intestines, such as fructans. Okay, so that leads us to the end of chapter 12. Let's go ahead and jump into chapter 13. All right, chapter 13, microbe-human interactions. Please know the difference between infection and disease. Remember that infection alone does not mean that you show the disease. Please know what the normal biota is. All right, please know where on your body, where are sites that are definitively known to harbor microbiota. So these are the sites where microbiota are normally found. And then be able to tell me where microbiota should not normally be found. Remember, those examples are inside of the tissues or inside of the blood or even in the lower respiratory tract. That's not a normal place to find microorganisms. Please know where your microbiota start. Remember, this occurs during birth, right? During birth is when you pick up your mother's microbiota. Okay, please know the difference between types of pathogens. First of all, know what a pathogen is. What is a true pathogen, which is a microbe capable of causing disease in healthy people, versus an opportunistic pathogen, which is a microbe that causes a disease in immunocompromised people and if introduced into parts of the body which is not natural to them. Please know what virulence means. Remember, this means the degree of pathogenicity, and then know what a virulence factor is. Remember, I told you that anything that helps a bacterium or a microorganism to cause a disease is called a virulence factor. Please know what an infectious dose is. Remember, it takes more than one microorganism to make you sick usually, right? It's the minimum number of microbes required for an infection to proceed. And remember that microbes with smaller infectious dose are usually ones that are more virulent. Please know the steps involved when a microbe causes disease. Remember, finding an entry portal, attaching firmly, surviving the host defenses, causing damage, exiting the host. Again, remember that virulence factors are any structures, products, or capabilities that allow a pathogen to cause infection in the host. Now, we talked a lot about toxins, so let's remember about toxins. Especially, I want you to recall the difference between exotoxins and endotoxins. So let's talk about it. Exotoxins are secreted by a living bacterial cell, and exotoxins, remember, are proteins that are heat labile, which means they are susceptible to heat. Endotoxin, remember, is not actively secreted by bacterium. In fact, it's part of the gram-negative outer cell membrane. Remember lipopolysaccharide, or LPS? Remember that lipopolysaccharide, the lipid portion of it is called lipid A, and that lipid A is endotoxin. Remember that. Again, what do I need you to remember? I need you to remember the difference between exotoxins and endotoxin. Specifically, I need you to remember these things, that exotoxins are unstable to heat, whereas endotoxin is stable to heat. That, again, I told you exotoxins are small proteins that are toxin. That means they are coded for by genes, whereas endotoxin is lipid A. Remember, it's the lipopolysaccharide of the outer membrane of gram-negative cells only. So they're only found in gram-negatives, whereas exotoxins can be found in gram-negatives and in gram-positives. And definitely, we spent some time talking about this, didn't we? The difference between a sign and a symptom. Remember, signs are objective evidence of a disease, something that could be measured or witnessed, whereas symptoms are subjective evidence of a disease. These are things sensed by the patient. So please know the difference between signs and symptoms. Here's a list of signs. Here's a list of symptoms. Remember, symptoms, you can't measure symptoms with devices. You need the patient to verbalize symptoms, whereas with signs, you can measure these things and witness these things and visualize these things. All right, and please know the stages in the course of an infection and disease. Remember the incubation period, the prodromal stage, the acute stage or period of illness, it's also called, and then convalescent stage. Please know what happens during each stage as well. See, please know what happens during each of these stages. And remember, can you be properly diagnosed during the prodromal stage? Let me see if you can beat Wicket on this one. Can you be properly diagnosed during this very, very early stage usually? That's right, Wicket. See Wicket chiming in with the answer. No, you normally can't be properly diagnosed. This is that morning when you wake up with the itchy throat, but it could be one of a dozen things you came down with. Does that make sense? So normally you need to wait until the period of illness or the acute phase of the disease to be properly diagnosed. So let's remember that. Please know the difference between a reservoir and a source. Remember, all reservoirs can be sources, but not all sources are reservoirs, right? Remember that? A reservoir is the primary habitat in the natural world where the pathogen originates, whereas the source is where you may have picked it up from. Please know the different carrier states, right? So know the difference between these types of disease carriers. All right, please know that vectors are living transmitters of disease. Normally, the majority of vectors are arthropods, these biting insects. Please know that zoonoses are diseases where they originate from animals. All right, and also please know vehicles, fomites, right? These are non-living transmitters of disease. Please know these terms as well. Please understand Koch's 4 postulates. Do you remember Koch used his postulates in order to link a particular disease to a particular microbe species? And please know his 4 postulates here. Postulate 1, find evidence of a particular microbe in every case of the disease. Postulate 2, isolate that microbe from an infected subject and cultivate it in culture, right? Isolated in culture, pure culture in the laboratory, perform full microscopic and biological characterization. Postulate number 3, inoculate a susceptible, healthy subject with the laboratory isolate, the bacterium, and observe the same resultant disease. And then lastly, postulate 4, re-isolate the same agent, the same microbe from that diseased animal, right? So please remember the 4 postulates. Remember, these were important because this is how Robert Koch showed us that a particular species causes a particular disease. All right, and that leads us to the end of chapter 13. This is great. We're well on our way. Let's take a quick break time with Gizmo and Wicket and see what they're up to. We'll be right back for the final 2 chapters. All right, welcome back from break time with Gizmo and Wicket. Let's go on to chapter 15, covering host defenses. Remember, we talked about 2 main branches of host defenses. Very important, the innate branch and the acquired branch. Remember, chapter 15 covers the innate branch mainly, and this is the branch of the immune system that does not improve with age. It does not have a memory. This includes the first line of defense and the second line of defense. And then chapter 16 covers the acquired or specific immune system. This is the branch that does have a memory, that does improve with each subsequent exposure. Remember that antigens are any molecule that can be identified by the immune system as either self or non-self. Remember that these antigens are commonly found on organisms, and the most common of these antigens are called pathogen-associated molecular patterns, or PAMPs, and these are recognized by receptors called pattern recognition receptors on your white blood cells. Remember, we spoke about the lymphatic system. Remember that lymph is a plasma-like liquid carried by the lymphatic circulation. Oh, and we talked about this very interesting fact. Remember that lymph moves in one direction only, from the extremities to the heart. So it's kind of like the circulatory system, but it only moves towards the heart. And the way that lymph moves is only through contraction of skeletal muscles, not through the beating of the heart. Remember, we talked about the lymphoid organs. The primary lymphatic organs are the red bone marrow and thymus. The secondary lymphatic organs are lymph nodes, spleen, and then the lymphoid tissues, salt, malt, and galt. Remember, the red bone marrow is very important because all blood cells originate in the red bone marrow, including the B and T lymphocyte precursors. Remember, the thymus was important. It's a triangular structure in the upper thoracic region, and it's where the naive T lymphocytes mature. So T cells mature in the thymus. Remember, the lymph nodes are so important to understand. These are small encapsulated bean-shaped organs, and their job is to filter out materials that have entered the lymph and to provide appropriate cells for immune reactions. This is where the B cells and T cells interact with foreign antigen. And remember that an enlarged lymph node is indicative of an infection. Remember that the spleen is also a lymphoid organ. The spleen serves as a filter for blood instead of lymph. Now, it has two functions. Its primary function is to remove worn out red blood cells from circulation. Its immunologic function is to filter pathogens from the blood for phagocytosis by macrophages. So again, not only are you filtering out red blood cells, but you are introducing any microbes in the blood to the phagocytic macrophages for clearing. Next, remember we talked about all the different types of blood. So you should know that the leukocytes means white blood cells, and you should know non-leukocytes include the red blood cells and the platelets. You should kind of know a few things about each of these leukocytes as well. So for example, here's what I want you to know. I want you to know that there are 3 phagocytic white blood cells. There are the macrophages, there are the dendritic cells, and there are the neutrophils. Those are phagocytic cells. Next, you should know that there are 3 antigen-presenting cells, or professional antigen-presenting cells. These are the macrophages, the dendritic cells, and the B cells. These are the professional APC cells, the professional antigen-presenting cells. These are the ones that possess MHC2. Next, what else do we need to know? We should know, again, neutrophils are phagocytic cells, but not professional antigen presenters. We should know that basophils, these have a function in inflammation. They can release histamine. And mast cells as well. Remember, mast cells play a role in inflammation. They can also release histamine. And then eosinophils, remember, they release anti-protozoal, anti-Helminth factors. Then we talked about the T cells. The T cells mature in the thymus, and the T cells, we'll discuss them more in chapter 16. And then the B cells, they mature in the bone marrow, and we'll talk about them more in chapter 16. Natural killer cells, these are interesting. They kill sick host cells. All right, you should know these different primary physical and chemical defense barriers against microorganisms. Remember, inflammatory response, we need to know these Latin terms: rubor, calor, tumor, dolor. So remember these terms: rubor, redness; calor, warmth; tumor, swelling; and dolor, pain. And then remember the 5th sign of inflammation: loss of function. Oh, remember the major events in inflammation? There's this damage that occurs. The damage triggers inflammation. Mast cells release their cytokines to open up the blood vessels, right? Do vasodilation, and by releasing histamine. This allows neutrophils to stop rolling on the blood vessel wall, called margination. Remember that term, margination? And then through diapedesis, these neutrophils squeeze through the blood vessel to get to the site of infection for phagocytosis. Remember all that? Again, you can see there's margination. So again, these pores open up in the blood vessels due to histamine from the mast cells. Margination is where the neutrophils line up on the blood vessel. They stop rolling and stick to the side of the blood vessel. Diapedesis, this means that the neutrophil squeezes through the blood vessel and then makes its way through chemotaxis to the site of infection to phagocytose those intruders. And remember, this all occurs during acute inflammatory response. Remember, we talked about fever as well. It's an abnormally elevated body temperature. Remember, there are ranges of low grade, moderate, and high, and that not all fever is bad. Remember how fever is triggered? There are these cytokines released by monocytes, neutrophils, and macrophages during the process of phagocytosis. And these cytokines make their way to the hypothalamus to reset the hypothalamic thermostat to a higher setting. Remember, there are benefits to fever, so please read and learn about these benefits of fever. Fever is not always bad. And remember, there are reasons to allow fever to play out, it's only when the fever is high and prolonged where it's risky and the fever should be treated. Otherwise, the fever is having a beneficial effect. And then, do you remember when we talked about another process here in the innate immune system called interferon? A virus infects a cell. This cell, if it realizes it's infected with a virus, this cell can activate the interferon gene to form the interferon protein. Interferon protein is a cytokine released by this cell that attaches to adjacent receptors on adjacent cells. Okay, and this adjacent cell will activate genes to synthesize antiviral proteins. Now remember, this adjacent cell, it's not protected from infection by virus, but it is protected against being taken over by that virus. And the infection ends at this cell. Does that make sense? So again, with interferon response, this is part of the innate immune system. This does not prevent the adjacent cells from becoming infected. However, it does stop the progression of the infection. And do you remember complement? Over 50 blood proteins, serum proteins that play a role in fighting off infections. All right, and that's it for the innate immune system. Please know that everything talked about in this chapter covered the innate immune system, the branch that does not improve with exposure, the branch that does not form a memory. So let's move on to our final chapter. All right, this is it, our final chapter, chapter 16. Let's see what we need to know. Again, please know what antigens are. Remember that B cells mature in the bone marrow while T cells mature in the thymus, and then they migrate to those lymphoid organs. Remember the lymph nodes, the spleen, and they establish a residence there. Remember MHC molecules, so important, MHC2, MHC1. Remember that MHC1 appears on all nucleated cells, including the antigen-presenting cells, the professionals. And remember MHC2, these are found on only the professional antigen-presenting cells. Remember the macrophages, dendritic cells, and the B cells. Remember that B cells have antibodies on their surface called B cell receptors. T cells have receptors on their surface called T cell receptors. They are both highly specific to their particular antigen that they recognize. So no two B cells recognize the same antigen. No two T cells recognize the same antigen because each one has a unique receptor. Remember, antibodies are made up of 2 heavy chains and 2 light chains, so 4 total proteins. And this is the tip of the Y is known as the variable region. This is the region that's specific to different antigens, that sticks to the antigens. And then the rest of it is called the constant region because it is not different. They're a constant, and they do not stick to antigens. All right, do you remember how MHC2 works? A professional antigen presenting cell will present antigen on MHC2, and it's the helper T cells, the CD4 positive helper T cells that recognize that foreign antigen on MHC2 on professional antigen presenting cells. This is what activates the helper T cells. And remember what the helper T cells do? They go and they help B cells become fully activated. Remember, and then fully activated B cells, they form memory B cells and plasma cells to fight the infection now and for later on, right? These memory cells remember that infection for next exposure. The B cells, the plasma cells that formed, they produce antibodies to fight the infection now. Okay, so I'm going to throw a card above me. If you look above me right now, there should be a card, and that card will show you the little video I had put together on how helper T cells become activated and how they move on to help the B cells become activated. So please watch that short video. It was also an extra credit assignment. I had you guys watch, those short series of videos of 5 videos. So you know what I'm talking about. Those videos will help you to understand how these T cells and B cells work. All right, and similarly with MHC1, remember even antigen presenting cells have MHC1. Antigen presented on MHC1 will be recognized by CD8 positive cytotoxic T cells. These become activated, they form memory cells, they become effector cells, and they can destroy any host cell, any one of your nucleated cells, any one of your host cells that also present that antigen. Remember that? So again, I'm going to throw a card up and you can see a video covering this topic as well. Remember, this was another one of the series of 5 videos you had seen earlier. Okay, and then lastly, I wanted you to remember this term opsonization. Remember that? Opsonization, this is when attachment of antibody to foreign cells and viruses exposes the epitopes to which they are bound to phagocytes. So remember that when an antibody is stuck to a foreign substance, such as a virus or a bacterial cell, the phagocytes, like the macrophages, dendritic cells, neutrophils, they have an easy time spotting these and eating them up, right? That's what opsonization refers to. All right, and that's it. We did it. We made it to the end. I hope this review helps. Again, please try to cover all the material because especially the ones we covered in class, because I may have missed, a thing or two, but I tried to cover all the important topics for you. I wish you luck. Please do your best. Let's finish this semester strong, and I'll catch you guys next time.
Dr. D, Dr. D, Dr. D. 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 Dr. D, Dr. D, Dr. D.